Hello, my name is Dr. Rob DeLong. I'm an associate professor in the Nanotechnology Innovation Center, Kansas State, otherwise known as NICS, in the Department of Anatomy and Physiology in the College of Veterinary Medicine. Today I'd like to overview our recently accepted article in the International Journal of Nanomedicine entitled Comparative Functional Dynamics Studies on the Enzyme nano-bio interface. So the basic overall question we were trying to address in this research report is a nanoparticle meets an enzyme, and there's a nano-bio interaction that occurs, and when that occurs, what's the functional impact on the enzyme? What happens to its biochemical activity? How do we characterize the biophysical interaction? To illustrate the dynamics of the nano-bio interaction, my co-author and colleague Dr. Comer performed a molecular dynamics simulation of the protein luciferase as it interacts with, or as it absorbs and migrates across the boron carbide surface. The representation of the protein emphasizes its secondary structure. Portions of the protein making contact with the surface you can see partially unfold during the simulation. Two things that really jumped out at us, or the two materials that really jumped out at us, were the boron carbide and the copper. There was a pretty significant change both in the intensity maximum, Imax, and in the change in area underneath the curve relative to the other nanomaterials, which are essentially physiologically based metal oxide materials that are known to have an active nano-bio interface, known to act or interact with proteins. And then the functional impact of the enzyme luciferase produces light, so we can use luminescence spectroscopy or bioluminescence from the enzyme as a measure of functionally how much does the nano-bio interaction of the different materials impact the activity of the enzyme. And here again, the boron carbide and the copper really stood out. In fact, in the presence of the boron carbide nanoparticle, we were unable to detect any activity. And in the presence of the copper nanoparticle, clearly the enzyme kinetics was changed pretty drastically, and as was the overall change in activity. Another way to characterize the impact of the nano-bio interaction on the protein is by running the protein through gel electrophoresis before and after the nanoparticle has been incubated with the protein, and then digesting the protein with an enzyme, a typical enzyme that's used for this is trypsin, and then analyzing the digest pattern on the gel after various times of nanoparticle exposure. And in this type of a comparative scenario, it was very clear again that the two nanoparticles have distinctive effects on the, in this case, the structure of the enzyme. And then finally, in looking at the structure of the protein by CD, or circular dichroism spectroscopy, which again is able to discriminate secondary structure features within the protein. For example, what percentage of the protein has an alpha helical structure, or what percentage of the protein has a beta pleated sheet structure, or what percentage is in so-called random coil, neither of those two structures. And here again, it was very clear that the presence of either the copper Cu nanoparticle or the boron carbide nanoparticle had a pretty dramatic effect on the two different, on the alpha helix or the beta pleated sheet in both cases. Again, indicating that the nano-bio interaction between these two types of nanoparticles in particular definitely has a structural as well as a functional impact on the protein, in this case again luciferase. In overall summary, these results point us to evaluating this nanoparticle or NP series against some other proteins that would be physiologically important or biomedically relevant enzymes.